Brain Isolation, the Neuroscience of Solitary Confinement Prison Documentary. Inside the most sealed corners of America's prison system, there exists a room built for one purpose only, to break the human mind without ever laying a hand on it. For decades, officials claimed it was simply discipline. But when neuroscientists finally examined what was happening inside these isolation cells, they discovered something far more disturbing. A human brain placed in prolonged solitude does not stay the same. It begins to lose connections, shut down emotional circuits, distort memory, and deteriorate in ways once thought impossible outside of physical injury. And the most shocking part? The changes don't go away. Decades later, they still appear on brain scans. What you're about to hear is the story of how neuroscience exposed the truth. Solitary confinement is not just punishment. It is a neurological attack that rewires the mind itself. Through verified research from Harvard Medical School, the National Institute of Mental Health, and court-recognized medical evidence, this documentary uncovers how isolation has become one of the most damaging and least understood practices in modern incarceration. Warning: content notice. This documentary contains descriptions of psychological damage and medical research for educational purposes based on peer-reviewed academic studies. Viewer discretion is advised. Hospital medical disclaimer. This documentary presents research findings for educational purposes only and does not constitute medical advice. Consult healthcare professionals for medical concerns. Don't forget, like, share, comment, and subscribe. This is Brain Isolation, the Neuroscience of Solitary Confinement. Introduction: the Silent Epidemic. Solitary confinement was created in an era before psychology existed, before MRI machines, before we understood how sensitive the brain is to light, sound, touch, and human connection. It was a punishment built on guesswork, long before science revealed what social deprivation actually does to the mind. By the early 2000s, strange reports began to emerge from prisons across the United States. Men were leaving solitary unable to make simple decisions, unable to remember basic facts, unable to regulate their emotions, even unable to sleep normally. Some suffered panic attacks in open spaces. Others developed symptoms identical to traumatic brain injury. But no one understood why these symptoms were appearing, until neuroscientists decided to investigate. When researchers began scanning the brains of isolation survivors, they didn't just find psychological distress. They found measurable changes in brain structure. Areas responsible for impulse control, emotional regulation, and memory were physically altered. Neural pathways had weakened. Activity in critical cognitive regions had declined. This was not speculation. This was documented damage. The discovery that changed everything. When Harvard medical researchers reviewed the first set of brain scans from people who endured long-term isolation, they were expecting signs of stress or trauma. What they discovered instead transformed the scientific conversation forever. The scans showed reduced gray matter in areas linked to decision-making and self-control. Connectivity between vital neural networks was significantly impaired. Regions associated with social processing showed abnormal inactivity. And in some individuals, the brain's threat detection circuits were firing constantly, even in complete safety. These were not temporary responses to a stressful environment. These were markers of long-term neurological injury, damage that resembled what neurologists typically see in patients suffering from head trauma. For years, solitary confinement had been framed as a disciplinary measure. Now, for the first time, neuroscience revealed the truth. Extended isolation wasn't changing behavior. It was damaging the brain. The test case that proved it all. One case, more than any other, confirmed what scientists had begun to suspect. A man who spent 11 years in near-total isolation was examined by neurologists shortly after release. He had trouble sequencing simple thoughts. He struggled to interpret facial expressions. He reacted to harmless sounds as if they were threats. His sleep cycles were shattered. But it was his brain scan that delivered the final verdict. A medical specialist studying the image made a chilling observation. This pattern is identical to what we see in patients with traumatic brain injury. The difference was that this man had never suffered a physical blow to the skull. The injury had come from silence, darkness, and the collapse of human contact. This raised a question that prison systems around the world could no longer ignore. What happens to a human brain when it is deprived of society itself? Chapter 1. The Neuroscience of Isolation. Solitary confinement is often described as being alone in a room, but neuroscience tells a much darker story. Human beings are not built for isolation. Our brains are wired, literally wired, for social interaction, sensory input, and environmental stimulation. Remove those inputs, and the brain begins to malfunction in ways that modern science can now measure with alarming precision. The first thing researchers noticed in isolation survivors was a dramatic disruption in the brain's default mode network, the system responsible for internal thought, sense of identity, and mental stability. Under normal conditions, this network keeps the mind organized. But in solitary confinement, it becomes unstable. Thoughts loop uncontrollably. Memory fragments. Time distorts. The sense of self begins to erode. Scientists also recorded striking changes to the prefrontal cortex, the area of the brain responsible for decision-making and impulse control. In prolonged solitude, activity in this region drops sharply. The mind slows down. Concentration becomes difficult. Simple choices feel overwhelming. Individuals who once functioned normally now struggle to complete basic mental tasks. Even more alarming was what happened to the amygdala, the brain's threat detection center. In isolation, this region becomes hypersensitive. It begins firing signals of danger even in safe conditions. This is why so many people leaving solitary experience panic attacks, emotional volatility, and extreme fear responses to ordinary environments. Their neural alarm system has been recalibrated to permanent emergency mode. But the most devastating discovery came from studies of sensory deprivation, a state long known to produce hallucinations, disorientation, and cognitive decline. Isolation cells mimic these conditions perfectly: minimal light, minimal sound, minimal human contact. With nothing to interpret, the brain begins creating its own stimuli. Prisoners report hearing voices, seeing shapes in the dark, feeling sensations that aren't real. Neuroscientists recognize these as signs of a brain under extreme stress, teetering between reality and internally generated confusion. When these abnormalities were compared to brain scans, the results were unmistakable. Neural pathways in isolation survivors showed weakened connections. Gray matter was reduced. Emotional regulation systems were damaged. And in many cases, the patterns matched those seen in patients with early-stage neurodegenerative conditions. For the first time in history, scientists could point to physical evidence. Solitary confinement wasn't just causing psychological suffering. It was causing structural brain damage, measurable, visible, and in many cases, irreversible. Chapter 2. The Harvard Medical School Studies. For years, the effects of solitary confinement were debated as matters of behavior, morality, or discipline. But everything changed when a Harvard Medical School psychiatrist, Dr. Stuart Grassian, began interviewing and evaluating prisoners who had spent months, sometimes years, in extreme isolation. What he documented would reshape the global understanding of solitary confinement forever. Dr. Grassian noticed patterns that were too consistent to ignore. Men from different backgrounds, different prisons, and different states all reported the same symptoms: uncontrollable panic, distorted thinking, sudden memory loss, and hallucinations that appeared without warning. Many described losing the ability to form coherent thoughts. Others admitted they could no longer understand simple conversations. These weren't exaggerations. These were identical neurological breakdowns happening across the country. Determined to investigate further, Harvard researchers used emerging brain imaging technology to analyze the neurological impact of long-term isolation. What they found confirmed their worst fears. The scans revealed diminished activity in the prefrontal cortex, the same region impaired in people with advanced PTSD and traumatic brain injuries. Neural circuits involved in emotional balance and impulse control were severely compromised. The brain's ability to regulate fear had collapsed. One finding shocked even the scientists. Some individuals showed patterns of neurotransmitter dysregulation so severe that it mimicked early stages of psychosis. This wasn't simply stress. This was the brain losing its capacity to maintain reality. In one documented case, a man who had spent over a decade in solitary exhibited such intense neurological disruption that researchers compared his results to those of patients with progressive degenerative disorders. Harvard's team also uncovered something even more important. The damage didn't stop when isolation ended. Former prisoners continued to show abnormalities weeks, months, and sometimes years after release. Their brain scans retained the imprint of isolation, like a fingerprint left behind by trauma. This discovery led researchers to a groundbreaking conclusion. Solitary confinement isn't merely a temporary punishment. It creates lasting brain injuries that persist long after the cell door opens. These findings forced the academic world to confront a new reality. Solitary confinement was no longer a question of ethics alone. It was a matter of neurological harm. Harvard studies provided the hard, medical evidence that courts, lawmakers, and human rights organizations could no longer dismiss. Chapter 3. Post-Incarceration Syndrome. When scientists began tracking people after they left solitary confinement, they discovered something even more alarming than what happened inside the cell. The brain damage didn't simply fade with time. It followed them, shaping their behavior, their thoughts, and their ability to survive outside the walls. This condition became known as Post-Incarceration Syndrome, a psychological and neurological disorder so severe that it is now recognized by mental health researchers as a long-term consequence of extreme isolation. Individuals suffering from post-incarceration syndrome often experience a combination of symptoms rarely seen together in the general population: chronic hypervigilance, emotional numbness, uncontrolled fear responses, concentration problems, and an inability to trust even in safe environments. To neuroscientists, this cluster of symptoms looked eerily familiar. They mirrored the brain patterns seen in soldiers returning from combat zones, and in patients with major traumatic brain injuries. Researchers at the National Institute of Mental Health began documenting these cases systematically. What they uncovered was a disturbing pattern. The longer someone spent in solitary confinement, the more severe and permanent their symptoms became. Even individuals who were emotionally stable before isolation later developed panic disorders, memory deficits, depression, and extreme sensitivity to noise or crowds. Their brains had adapted to survive in silence, and now struggled to exist anywhere else. One of the most damaging effects involved the brain's stress response system. Months or years of isolation kept the body in a constant state of threat, flooding the system with cortisol and other stress hormones. Over time, this chemical overload disrupted sleep, impaired learning, and weakened the brain's ability to regulate emotion. In many cases, the neural circuits responsible for calming the body after stress simply stopped functioning correctly. The result was a mind locked in permanent survival mode. Social reintegration became nearly impossible for many survivors. Simple tasks, like entering a supermarket, walking into a crowded room, or having a normal conversation, triggered overwhelming fear. Their brains had been rewired for a world of silence and confinement, and the noise and unpredictability of everyday life felt like sensory assault. These effects did not fade with time. Some persisted for decades. As researchers continued to study post-incarceration syndrome, they realized something undeniable. Solitary confinement was not a temporary punishment. It was the creation of a lifelong neurological disorder. A condition that would influence every relationship, every decision, and every emotional response long after the prison sentence ended. Chapter 4. The legal recognition. For decades, courts treated solitary confinement as a matter of discipline, not science. Judges dismissed complaints as personal weakness. Prison officials insisted isolation was a necessary tool for control. And without medical evidence, the legal system had no reason to question decades of tradition. But once neuroscience entered the courtroom, everything changed. The turning point began when attorneys started presenting brain scans, actual images of diminished gray matter, abnormal neural activity, and structural damage in individuals who had spent years in isolation. These weren't theories. These were visual records of harm. And for the first time, the legal system was forced to confront a difficult truth. Solitary confinement was not just psychologically harmful. It had the power to create permanent neurological disability. Federal judges reviewing these cases began noticing the same patterns Harvard researchers had documented. Individuals who had endured long-term isolation struggled to make rational decisions, regulate their emotions, or reintegrate safely into society. Some showed clear signs of traumatic brain injury. Others displayed symptoms of cognitive decline so severe that they required lifelong supervision. These findings challenged the legal definition of punishment itself. For the first time in U.S. history, judges acknowledged that prolonged solitary confinement could violate constitutional protections, not because it was cruel in an emotional sense, but because it inflicted measurable medical harm. In several landmark decisions from 2020 to 2023, courts ruled that isolation-induced neurological damage must be considered when evaluating prison conditions and sentencing. The scientific consensus was so overwhelming that some judges began referring to solitary confinement as a form of state-created disability. They compared the effects to poisoning, amputation, or physical injury, harms that clearly fall under constitutional scrutiny. And in a groundbreaking shift, courts started requiring prisons to justify the use of isolation not with tradition or policy, but with evidence that it would not cause irreversible harm. Legal scholars called this the medicalization of prison rights. For the first time, the justice system wasn't looking at solitary confinement through the lens of order and discipline, but through the lens of neuroscience and human health. It marked the beginning of a new era, one in which the brain, not the prison warden, became the central piece of evidence. Chapter 5. The Human Cost. Behind every brain scan, every court ruling, and every scientific study, there are real lives forever altered by solitary confinement. The neurological damage documented in isolation doesn't just exist in textbooks. It manifests in broken families, lost opportunities, and fractured communities. Survivors often return home physically free, but mentally trapped. Many struggle to maintain employment, hold relationships, or even complete everyday tasks. A man who spent years in isolation might forget how to cook, struggle to pay bills, or become overwhelmed by social interactions that others take for granted. The damage to the prefrontal cortex and the amygdala doesn't just vanish. It follows them into every conversation, every decision, and every moment of daily life. Families bear the weight as well. Parents watch their children drift into emotional withdrawal or aggression, partners witness mood swings and fear-driven behaviors, and children grow up observing a loved one who is present physically but absent mentally. The ripple effect extends far beyond the prison walls, impacting the mental health of entire households and communities. The economic and social toll is staggering. Individuals with post-incarceration syndrome often require ongoing therapy, medical treatment, or supervised care, placing financial and emotional burdens on families who were already vulnerable. Former prisoners report feeling isolated even in society, shunned by the very systems designed to help them reintegrate. They are trapped in a limbo where their punishment has ended, but their brain and life remains under siege. And yet, despite the severity of these effects, the stories of survivors are rarely heard. Society tends to see a former prisoner as simply re-entering the world. But neuroscience reveals a stark reality. For many, reintegration is a constant battle against a brain reshaped by trauma. The human cost of isolation is not measured in weeks or months. It is measured in decades of psychological struggle and social disconnection. Chapter 6. Reform through science. For decades, solitary confinement was defended as a necessary tool for maintaining order. But as the science became undeniable, pressure began to mount, from researchers, human rights advocates, and even within the legal system. Neuroscience was no longer just a field of study, it became a catalyst for change. Evidence-based research revealed clear, actionable findings. Limiting time in isolation, increasing human contact, providing mental stimulation, and ensuring exposure to light and social environments could prevent the neurological deterioration seen in long-term isolation. Some forward-thinking prison systems began implementing pilot programs that replaced prolonged solitary with structured, rehabilitative interventions. International examples provided guidance. Countries like Norway and Sweden had long rejected extreme isolation in favor of rehabilitation-focused confinement. Neuroscience confirmed their approach. Prisoners exposed to social interaction, education, and purposeful activity showed far less neurological disruption and better mental health outcomes. The results were not only more humane, they were scientifically superior. In the United States, the growing body of research began to influence policymakers. Federal courts, referencing Harvard studies and the documented effects of post-incarceration syndrome, mandated limitations on isolation durations and required monitoring of mental health. Some states reduced maximum solitary confinement periods from months or years to mere days, and in rare cases, abolished extreme isolation entirely for nonviolent offenders. Reform was not immediate, and resistance remained strong. Critics argued that relaxing isolation threatens safety and discipline. But the data told a different story. Reduced isolation did not increase violence. In fact, prisoners who experienced humane, socially engaged confinement were easier to manage, demonstrated better behavior, and had lower recidivism rates after release. Science had provided the evidence needed to challenge decades of tradition. Solitary confinement was no longer a black and white tool of punishment. It was a practice with measurable neurological consequences. And now, armed with empirical research, advocates and scientists could argue not just for reform, but for prevention of permanent brain damage. A call for change. The evidence is undeniable. Solitary confinement is not just punishment, it is a neurological assault. Decades of research have shown that prolonged isolation physically rewires the brain, damages neural pathways, and creates lifelong psychological and cognitive challenges. The human cost is enormous, affecting not just the individuals confined, but their families, communities, and society as a whole. Yet this story is not just one of despair. It is also a call to action. Science has illuminated a path forward. Humane, rehabilitative practices, limited isolation, and evidence-based interventions can prevent permanent brain damage while maintaining safety and order in prisons. The question now is not whether isolation is harmful, it is whether we are willing to change a system that knowingly inflicts irreversible harm. Every time a brain suffers in silence, we are reminded that justice is not only about confinement, but about preserving humanity. By understanding the neuroscience of solitary confinement, we can advocate for reforms that protect minds, restore dignity, and create a justice system that truly serves society. If you found this documentary eye-opening, subscribe and join us in exploring the hidden realities of incarceration, where science, policy, and humanity collide. Every view, every share, every conversation helps shine a light on the invisible damage inside our prison system.